Match question. F1 particles, this is associated with mitochondria. So A is 4. A4 is in 2, 3, 4. So B is either 1 or 3. Histones is chromosome. So B is 1. And now we have to rule out between C2 and C3. Axoneme is structure in cilia. So it is 2. So our answer is option number 2. So mark it on the OMR sheet, and this will be option 2. So now note down the time. This is the time you took to attempt this question. So bear in mind, you will not have a timer in the actual exam, and you don't have watch also. So while you are practicing, this is your preparation phase, and when you are practicing, try to feel the time, the passage of time, and how much is 1 minute, how much 1 minute feels. There will be a lot of tension in that exam hall, and it is all about how quickly you can solve your question. So let's understand all these terms asked in this question. First one, F1 particle, it is associated with mitochondria. So remember, mitochondria it's a double membrane organelle, and the inner membrane has infoldings. So this is called cristae. Okay, don't get confused with cisternae. Cisternae is for Golgi, and this is cristae. So you can relate it to Christ. God gives you power, and cristae is associated with an organelle which also gives power. Okay, so this is one trick you can use to relate and remember what is F1 particle. So if you zoom into the membrane of mitochondria, inner mitochondrial membrane, it will have one structure which is in the membrane. Okay, this is called F0 subunit. Then there is a stalk, okay, and then outer unit which is called F1. So this part is the intermembrane space, and this is the mitochondrial matrix. So there are a lot of protons over here. Okay, this is flooded with protons. So when the protons they come out into the matrix, they cause rotation, okay, in this part, and this rotation is coupled to ATP. They can even ask you F0, and this is the channel, and the proton will bind to F0 subunit. So next one, histone. What are histones? Histones are basically, they are like the spool around which the thread is wrapped. So histones, they are basically octamer proteins. They are positively charged protein. And what is the charge on DNA? DNA is negatively charged. So imagine there are 4 more structures, spherical globular proteins. Okay, so together they form octamer, 8 proteins, histone proteins. So DNA is wrapped around it. Okay, wrapped around it, and this is how it doesn't get entangled. So this one structure will be called nucleosome. There will be another histone which is called H1. It is the linker histone, and these are H2A, H2B, then H3 and H4. So they form the octamer, so cilia and flagella. So you have the structure, extended structure. the internal strength is provided by the microtubule filament, which is filled inside. Okay, so this is going to give the strength and the function to cilia and flagella. What is it made up of? Microtubules. So the basal body, this will have the centrioles, and centrioles help in the polymerization and formation of the internal microtubule filament. So exoneme, if they ask you which cytoskeleton and is associated with this, you have to write microtubules. Coming to the last point, the Golgi. So this will be like this, convex shaped. Okay, and then you will have other sac like structures, and this will be concave in shape, the end which faces the plasma membrane. So this is called the cis phase, or this is the growing phase, and this one is the trans phase, which is called the, and it will be secreted out into the via the plasma membrane into the extracellular space. So cisternae are these flattened sacs, and two types which you need to remember is cis phase and trans phase. Cis phase is the growing phase, and trans phase is the maturing phase of Golgi apparatus. So that's all you have to remember with respect to this question.